Well, hello everyone. Today we're going to continue the lecture on the anterior pituitary from chapter 3, and then talk about the hypothalamus from chapter 4. I hope I have that sequence correct, but we'll work our way through it. So I'm going to first, go to our WebCT page and show you a link that is excellent for helping to understand some of the concepts, at least the final concept made in today's lecture. So under course materials, the course content icon, number 3, the HPA-HBG axis interactions. There is an article, a link to an article there. It's very interesting very interesting discussion on the interactions between the hypothalamic pituitary adrenal axis and the female reproductive system. And you'll see that I've pulled at least one slide, I think it's this figure here, and possibly another one from this. It's a very interesting article. It helps us to understand many of the aspects of interactions of the different neuroendocrine axes, as well as expanding our understanding of how the human reproductive endocrine system can be affected by the environment. So we continue then with the discussion of the anterior pituitary gland, talking about the neuroendocrine axes associated with it, and of course the hypothalamus, completing the lecture series on chapters 3 and 4 from the textbook. You will also find a very nice discussion and description of the pituitary gland at the endotext link on our course website. So these are glandular cells. We're becoming very familiar with that now, and it has a rich capillary bed, something that we're even more familiar with, and we'll point it out as we go along. First, we want to remind ourselves of the two components of any organ. I'll just use my pen for the camera system here. The two components of any organ are its stroma and parenchyma. Let me zoom in on that. The other components, obviously, vasculature, and then a connective tissue capsule. So every organ will have a connective tissue capsule. The stroma is the connective tissue, CT is obviously the abbreviation for connective tissue, framework. So if we want to just scooch over and sketch it, we'd have a connective tissue capsule around the pituitary. Probably isn't very thick, but it is significant. And then there would be fibroblasts that will contribute to the connective tissue makeup of this, mostly collagen fibers and things like that. And you notice that I'm making these kind of circular almost. We talked about an acinus with reference to the secretory portion, the glandular portion of the breast tissue. If this ends with an I, of course, it's plural, so an acinine. What we'll see is that the parenchymal cells, these are the functional cells of the organ. Thinking of the particular function of the organ, we know all the cells within the in any organ have a function, but when we think of the organ having a function, like the heart, it's the muscle cells, the cardiac muscle cells that we think of as the parenchyma, and a tremendous connective tissue stroma for that as well. So the anterior pituitary is described as having a pseudoacinar structure. So a typical acinus is going to usually find that arrangement of glandular cells with exocrine glands. So they open into a duct and the duct opens onto the external surface of the body like a sweat gland or a digestive enzyme secreting gland of the pancreas. So definitely not for endocrine tissues, but for some reason, with the capillary being central and the parenchymal cells surrounding these, not always perfectly surrounding them as it would be with a true acinus. Those are the dots obviously are the nuclei of the cells, parenchymal cells. It ends up looking very much like a cross-section of an acinus. If we had seen the glandular tissue of the breast cut in cross-section, it would have a very similar arrangement as with a salivary gland or any other type of gland. So we'll see that now as we proceed through these lectures, through these next slides on the lecture of the structure of the anterior pituitary. We're going to focus on the cytology, particularly the parenchymal cells. So that seems obvious, but I just wanted to state it since we just distinguished between them. So what are the characteristics of the secretory cells, and as well a little bit on the nomenclature. So we're not going into great detail on these, but we are going to touch on them so we understand, at least from the historical perspective, the terminology for these cells and the ways of characterizing them. So in the nomenclature, that can be named based upon the staining characteristics, such as loving, color-producing stains, so chromophils, and these are parenchymal cells that have many granules within them. Then there's chromophobes, which obviously hate the stain, so they're going to be lightly stained or not stained at all, and they have few granules. So obviously the stain that's being used to view these parenchymal cells and end up with this nomenclature is targeting something within the granules. And they also have nomenclature for the hormones synthesized and secreted, such as the lactotrophs. There will be two approaches to the terminology that we will use for this. And we'll use the lactotrope. I usually call them, use the TROPE suffix rather than the TROPH as you saw in the slide. So lactotrophic cells, lactotropic cells, and obviously the lacto, the prefix, is telling us that these are prolactin synthesizing and secreting hormones. So basically the prefix is going to change. So the somatotrope will be the cell that synthesizes and secretes growth hormone, or somatotrophic hormone. And the corticotropes will be for the ACTH. The gonadotrophs, gonadotrope cell, obviously for the gonadotropins. There are two gonadotropins, LH and FSH, as we know. So what have we got left? Lactotropes, somatotropes, corticotropes. Well, we'll name them as we go along if more of them pop up. So I'll return to the slides, my cheat sheet. This slide is showing us three images, upper left, the right-hand side, and then the lower left. And the upper right-hand side is a low magnification histological section of the human pituitary gland that is stained. I'm not sure exactly what stain it is. I gave the address for the site from which I retrieved these images. It's really a cool site. I encourage you to go there and look at the way it's structured and some of the information that they have. I've borrowed some of the pertinent slides for our presentations. So we can see this glandular appearance. You can almost see the little capillaries in there, and that there's some kind of circular staining that surrounds them. That's that pseudo-acinar appearance, which certainly is obvious here in this higher magnification in the lower left-hand panel with the capillary in the center as I had drawn. And then the dots are the nuclei of the parenchymal cells. Here we see connective tissue cells, fibroblasts, contributing to the connective tissue stroma of the organ. On the right-hand side, well, first, sorry, hit them in the right location here. What we note first, obviously, is this is the glandular portion, the adenohypophysis. This doesn't seem to be staining really very much at all, which is kind of typical of nerve tissue. So this is the neurolobe. So Rathke's pouch, there's just a slight remnant here, and you probably would have to look at the slide very closely on the actual image on the computer. But I think you can see a little red strip here, and then a white strip to the left of it, clear so that there's nothing there. Obviously, the fluid content of Rathke's pouch, the cavity of Rathke's pouch, would be there, and then a red tissue on the other side. So there really isn't much pars intermedia left in association with the cavity of Rathke's pouch. But the pars intermedia cells do become incorporated into this strip just between the pars distalis and the neurolobe, the neurohypophysis. So I point that out because when we come to this panel on the right, we see that histologically with the processing, there's an artifactual separation of the neurolobe from the pars distalis. So we don't want to be deceived by that. But what we should be able to determine from this slide is that if this was the cavity of Rathke's pouch, there would have to be anterior pituitary glandular cells, the neural intermediate lobe, the intermediate lobe over here. So that's just an artifactual separation of the tissue with the processing. Here we see these colloidal cysts that persist in the human anterior pituitary gland, and basically these are little remnants of the cavity of Rathke's pouch. So this is the neurointermediate lobe. Why do I keep saying neuro? This is the intermediate lobe, pars intermedia, and pars distalis on the other side. And we still can see kind of these cords of cells around capillaries, when they're not cut in perfect cross-section, maybe along the parallel axis of the capillary, in other areas where it's just kind of clustering into these circular appearances. So that's that pseudo-acinar appearance that is most obvious at this high magnification shown here. So this would be the basal membrane of the parenchymal cell, the apical membrane, and the release is most likely to be at the apical membrane into the interstitial fluid where the capillary lies nearby. We'll proceed to a line drawing to illustrate the details of that ultrastructure in just a moment. But first, let's look at another type of a stain that actually shows the three different types of cells. The pars distalis is shown here, and the three major types of secretory epithelial cells are shown. The A is labeling the pale one, so one of the chromophobes. So there's one type of chromophobe, and then there's two types of chromophils. There's a blue basophil B. I don't see a lot of blue in that, but I guess that's my own eyes maybe. And then C is the red acidophil. So early on, the nomenclature arose from these types of classical stainings, and as methods developed, especially using antibodies against the hormones themselves to visualize them with other types of chromogens, their greater detail was generated. And this terminology really isn't used much anymore because we simply use the terminology that I had written, the somatotrope to gonadotrope and so on and so forth. So this is the legend describing that. Notice that the cells are characteristically cord-like clumps. The clumps are separated by fine reticular fiber stroma. There are thin blue lines. The blood capillaries lie there. And all the endocrine glands are highly vascularized, we know that. And the secretion goes directly, I left this here because we know it's better than that. It's not directly into the capillaries, it's directly into the interstitial fluid and washed away, diffused down its concentration gradient across the capillary wall, and then is washed away by the plasma within the capillaries. So I remind everyone that we go for details as much as possible in the course. And the endothelium of the vessels is typically continuous and fenestrated, and we can see that in this illustration. So I'll start first with the capillary here. This is a line drawing illustration of a high magnification, or medium to high magnification, as would be seen at the level of the electron microscope. And so let's walk our way through this, and then I'll read the figure legend that corresponds to these numbers. Let me find them. Where's 1? Oh, back there. 1 through 7. So we're drawing our attention first to the capillary here. We see the endothelial cell, and it has a tight junction where the edges of the cytoplasmic membranes come together. There's the nucleus and the mitochondria. Out here, just outside of it, is the Peri Capillary space. So there's fibers here, and here's a fibroblast. So those are the parenchyma and stroma and stromal cells. Hopefully you were correcting me immediately there too. And what we notice that there's 2 types of parenchymal cells represented: lactotroph or prolactin cell, and a somatotrope or a growth hormone synthesizing and secreting cell. So let's go back to the capillary for a moment. We see that there's no discontinuities. Here's another tight junction. So there's no blatant openings through the or discontinuous portions of the endothelial cytoplasm. So it's continuous. And we see the fenestra here. These are little windows, and they are selective for size, and the hypothalamic hormones can diffuse through those. So now we're saddling up on a hypothalamic hormone again, and we have come from the primary portal plexus through the portal vessels. Now we're into the secondary portal plexus and the pars distalis, floating around in the plasma here, and we diffuse down our concentration gradient into the interstitial fluid, bouncing around, bouncing around. If I am growth hormone releasing hormone, it's not going to do me much good to bounce into this prolactin cell unless it expresses the growth hormone receptor, growth hormone releasing hormone receptor. But if we happen to diffuse over this way or exit the vessel over here and bounce around, we might then bind to the growth hormone releasing hormone receptor on the somatotrope and trigger the intracellular signaling cascade after transmembrane signal transduction, trigger the intracellular signaling cascade required to cause exocytosis and the secretion of the growth hormone that would enter the interstitial fluid compartment and then diffuse down its concentration gradient across the endothelial cell, lining the capillary into the plasma and be washed away. At the same time, what we have to begin to picture in our minds is that intracellular signaling cascade somehow has got to get information to the nucleus, to the growth hormone gene, to increase gene expression such that there's new messenger RNA for growth hormone sent to the cytoplasm to produce the growth hormone in replacement of what was just released. Well, let's shift then over. Well, there's other cell types as well. There's a follicular cell that's kind of a strange cell. They probably know more about it now than the last time I checked the literature, but it's of no great consequence to us at this moment. Let's just focus on the lactotrope down here and start with number 1 and proceed to number 7 since we just talked about the synthesis of the anterior pituitary hormones. So somewhere in the nucleus, there's the gene for the corresponding hormone, in this case prolactin. It has upstream portion, the promoter region, with certain types of regulatory elements, response elements. regulating its tissue-specific and cell-specific gene expression, and then the modulation of the constitutive gene expression, increasing or decreasing with negative feedback, or to come, in order to come back to baseline, or increasing if there's an additional stimulus. So the messenger RNA comes out associated with ribosomes, starts translation at the rough ER, as we know. There might be some post-translational processing as this proceeds. I think on the recording the number 1 is off the screen, but that's fine. You can look at it on your printout. Number 2 is showing us the transport vesicles from the rough ER to the Golgi apparatus, as we know really well. There's additional post-translational processing and packaging. Number 3 here in the Golgi apparatus, production of vesicles from the Golgi apparatus that can fuse together to form large granules, secretory granules in these cells, those granules that stained in the chromophylls. And then these proceed. Again, there's intracellular mechanisms, definitely cytoskeleton, because any change in shape or movement within a cell of an organelle is going to require cytoskeletal involvement. So the cytoskeleton somehow is being told to migrate these in these patterns as we're seeing. And then the approximation, there's a signaling mechanism that will review on the approach, the docking, the fusion of the membrane to form this omega figure here. It looks like an omega, it's upside down right now, but once that omega figure appears, then the intravesicular compartment is open to the interstitial compartment, and the contents diffuses down its concentration gradient into the interstitial fluid, and so on and so forth. So there are steps, there is potential for numerous different types of post-translational processing of the hormone, from the pre-pro hormone back in the rough ER to the pro-hormone, and then pro-hormone maybe with some glycosylation or the guidance to form the disulfide bonds, as we talked about last time, through the Golgi, and sometimes even additional cleavage and processing through the vesicle to granule stage before secreting the biologically active, fully mature hormone into the interstitial fluid compartment. There is a life cycle for all molecules. If basal synthesis is slightly above what is required to have a readily releasable pool, anytime you have a readily releasable pool, some of it's going to age before it might be used, is a theoretical idea anyway. So if it sits around too long, it'll be tagged, and for example, ubiquitin is one of the tagging molecules that tells the cell that a vesicle should be shuttled to join with the primary lysosome, forming a secondary lysosome, and then the secretory granule is exposed inside the secondary lysosome to all those degradative and corrosive enzymes, and it is destroyed. So the life cycle of the molecule is synthesis, with processing, storage, activation to become biologically active, and then to have its biological activity and to be destroyed. If it sits in the reserve pool, in the inactive form, waiting to be activated too long, it can be destroyed as well. So we'll talk about the life cycle of a molecule a bit more in a subsequent lecture. But we have to keep that in mind. And then this figure legend, I won't read it for you, goes through the description that we just did. This slide is showing two electron micrographs. On the left is a somatotroph. On the right is a mammotroph. So these are actual electron micrographs. We see that the somatotroph has many granules, and it's the typical nucleus, rich in the rough ER and the Golgi apparatus. And then we see the somatotroph is not quite so filled with the the mammotroph with the secretory granules, but all the ultrastructural machinery is present to follow the procedure that we just described in the outlined illustration. Now let's continue on with our discussion of the adenohypophysis, describing the model system, which is a 3-gland, 3-hormone model system. So we're going to be talking about gland 1, hormone 1, gland 2, hormone 2, gland 3, hormone 3, and as well, in that context, we will describe the different anterior pituitary neuroendocrine axes with reference to the stimulus or stimuli, response, and regulation systems. So this is where we get into this loop of stimulus, response, and regulation, and we tend to forget the target cells of the hormones and the actual function of those hormones, but we'll do everything we can as we get into the details of each axis later to focus on that as well. Here's another illustration of this, building in the feedback loops. So hypothalamus is going to be glandular tissue 1, so it's not a real gland, but anyway, we can describe it that way. Hormone 1 will come from it. Anterior pituitary is gland 2 with hormone 2. Peripheral endocrine gland, if it exists, it doesn't in every case. With the hormone 3, and then the effector cells, hormone or hormones 3 will have negative feedback through the long loop at the gland 2 and gland 1. Short loop negative feedback is from hormone 2 to gland 1. There's even an ultra short loop negative feedback where hormones 1 can impact gland 1. So while we mentioned the feedback mechanism, let's look at this model. We have to have a controlled system that has an output and some way of disturbing it, which could be a stimulus. So we have to move it away from its static level, its baseline level. So the set point is contained within the control system, and the sensor, the set point is contained within the sensor system. Sorry about that. And the output of the controlled system impacts the sensor, and then there is a comparator function within the sensor that compares the level of output to the set point, and then there is an inverse amplifier that provides the feedback signal of the magnitude in the opposite direction of output to bring it back to the set point. So in other words, if we're at basal level and suddenly a disturbance increases the output by 10, then the comparator determines that 10 and makes it a negative 10, and the feedback says bring it back down by 10 to get back to baseline. Okay. So this obviously is a negative feedback model. So the inverse amplifier is essential in negative feedback, and there's just an amplifier in a positive feedback. So the output drives the system through some means to increase more and more until the stimulus to the disturbance is removed to terminate it. So this slide is asking us the question, what does negative feedback look like? For the steroids, for the hormone or hormones, 3 types of steroids. Steroid hormones, must be Friday. For steroid hormones, as hormone or hormones 3, it's freely diffusible from the plasma in the anterior pituitary. We saw the capillary there, right through the plasma membrane, through the nuclear membrane and nuclear envelope, binding to its receptor, and it will, once ligating its receptor, bind to the corresponding response or regulatory element on the target genes and decrease gene expression to bring it back to basal levels. So basal levels of messenger RNA would be recovered in the plasma, and the storage would be returned to the baseline capacity. If it is a water-soluble hormone from any particular gland, then it will bind to a membrane receptor, activate some type of intracellular signaling mechanism through transmembrane signal transduction, and get to the gene through the nucleus by altering some other type of transcription factor. In the case of the steroids, the receptor is a transcription factor. Any protein that binds to DNA and changes gene expression is a transcription factor. So the water-soluble hormones are going to have to activate intracellular cascades that ultimately will activate a transcription factor. In the case of this example, the cyclic AMP, there is a cyclic AMP response element binding protein, or CREB, and cyclic AMP will activate a protein kinase that will phosphorylate that CREB, and we'll talk about that in more detail later. And then once the CREB is phosphorylated, it has permission to go to the nucleus and can bind to the CRE, the cyclic AMP response element, on the target genes. And in the case of a negative feedback, it would decrease gene expression. In the case of hypothalamic releasing hormones that bind and stimulate release, the pathway splits to target synthesis, so it would stimulate exocytosis in one pathway, and also increase gene expression through the CRE response element. This is actually showing glucagon affecting the target cell, so the text in here doesn't fit the description that we're doing, but we should be able to supersede all of that because we're at such high level of functioning in our biology background. This slide makes the note that we're looking at the neuroendocrine axes of the adenohypophysis now. So this is the anterior pituitary, the adenohypophysis, the posterior pituitary, and the pituitary stalk. So gland 1 will be the hypothalamus. Hormones 1 will come down through the portal vascular system and bathe the target cells in the anterior pituitary. So it's always going to be hypothalamo-pituitary axis. And then the third name of the axis will be based upon the target organs. So there will be the hypothalamo-pituitary thyroid axis, hypothalamo-pituitary breast axis, because there isn't a third gland that secretes a hormone. Prolactin is going to stimulate or promote lactation, stimulate the secretory cells of the acini and the mammary glands. Breast milk is not a hormone, so there's no hormones 3. So this is what I said. In some cases like this, we know there's hormone 3 from the thyroid, so we've got 3-gland, 3-hormone model working perfectly. For the hypothalamo-pituitary breast axis, it isn't quite that way. And then for the adrenal gland, hypothalamo-pituitary adrenal axis. So these are abbreviated as HPT, HPB, HPA. And then for the gonads, the two gonadotropins shown here, the HPG, gonadal axis. And then the liver is involved with growth hormones, so there's going to be an HPL axis, and I challenge you to try to determine if there is an axis missing from this, and of course there is. There is the hypothalamo-pituitary skin axis for the alpha MSH that is synthesized and secreted by the parenchymal cells of the intermediate lobe. The melanotropes. I knew there was one missing. So thyrotropes in the anterior pituitary are involved in the HPT axis. Lactotropes are involved in the HPB axis. Corticotropes are involved in the HPA axis. And gonadotropes for HPG somatotropes for HPL, for growth hormone, and melanotropes for the HPS, the hypothalamic pituitary skin, which we don't deal with in any great detail in our studies. So here again we see this with the second gland, second hormones, and the target tissues, target organs, target tissues, target cells. So the ACTH, adrenocorticotropic hormone, is synthesized and secreted by the corticotropes in the anterior pituitary, targeting the adrenal cortical cells. Thyroid stimulating hormone is synthesized and secreted by thyrotropes. That was one we missed too, yeah. And targets cells in the thyroid gland. The gonadotropins are of two types, and there's one or maybe two populations of gonadotropes. There definitely are two gonadotropins, follicle-stimulating hormone and luteinizing hormone, each of which act on particular target cells within the male and female gonads, and we'll differentiate those as we proceed. The prolactin by lactotropes and targeting the glandular tissue, and we'll look at some details here in a moment. Somatotropic hormone, or growth hormone from the somatotropes. You got it touching the posterior pituitary, but we know it's coming from the anterior pituitary as labeled. And the growth promoting effects on most cells, and as well there's an effect on liver cells to secrete a third type of a hormone, and we'll discuss that in a bit of detail. So we have seen this slide one time before for oxytocin with the posterior pituitary, targeting the myoepithelial cells and the muscle cells in the wall of the ducts. Now we're picturing prolactin diffusing out of a capillary nearby, bouncing into everything, going anywhere that the oxytocin might also, but it happens to bump into and bind to its receptor on the epithelial milk-secreting cells, and through transmembrane signal transduction, activates or inactivates, whatever it might be, corresponding intracellular signaling mechanisms that activate the production of milk. We know there's a lipid content and a protein content and a carbohydrate content. So many cascades of enzymes being activated, and since it is a holocrine type of secretion, the entire cell fills with the milk substance, and then it is secreted itself as the whole cell breaks down and then is delivered out with the milk letdown reflex, as we know, dependent upon oxytocin. So maybe we do have to have gene expression as well, because the reserve enzymes for all of that synthetic capabilities might not be enough upon activation to complete the task, so it may require additional gene expression for those proteins, those synthetic enzymes, to complete the task of producing all of the milk that this cell will contribute to the total breast milk. So we always think about that. Will there be intracellular signaling? Most likely there will be, to generate the response. And will that response require gene expression or an alteration of gene expression up or down? Questions we always want to ask when we're trying to envision these. So obviously the thyroid gland is the target. We see the arteries here, so ultimately from the pituitary gland into the cavernous venous sinus, ultimately right heart, right atrium, right ventricle, through the lungs, through the left atrium, through the left ventricle, through the aorta, finally on the thyroid artery, coming into the thyroid. So this thyroid stimulating hormone from the pituitary gland is taking a long trip. I'm getting tired just describing it. And then diffuse out of the capillary and target the cells there, and we will go into details on that later. There are two thyroid glands. They're quite large, massive amount of tissue. The anterior pituitary has to produce thyroid stimulating hormone concentrations in the plasma after that whole route at a significant enough level such that the thyroid gland can be uniformly stimulated as needed. The male gonad is shown here, obviously. We recognize the many structures, seminiferous tubules, for sperm production, and the duct system that takes the sperm away from there to be joined by the liquid portions from the accessory glands to form the full components of semen. And we zoom in on the seminiferous tubule within this to remind ourselves of the target cells of gonadotropes. Here's the capillary scattered around, and then the connective tissue capsule around a seminiferous tubule, and we see the parenchymal cells. There's not a significant amount of stroma inside, but a very thick capsule outside. In the other stroma of the male gonad, the testes, there are some endocrine cells scattered. These are called the interstitial cells of Leydig, as we know, or Leydig cells. That's where the capillaries are. So now we're picturing we've saddled up on a gonadotropin. We diffuse out into the interstitial fluid, bouncing around. If we're on LH, we might eventually bounce and bump into our receptor on a Leydig cell and trigger a testosterone synthesis cascade inside that cell, enzyme dependent. Or if we're follicle stimulating hormone, we might get through that connective tissue capsule and bind to the FSH receptor on the basal membrane of the sustentacular cell, the Sertoli cell, and activate intracellular cascade such that we promote sperm development. And of course, the testosterone is going to diffuse away as soon as it's synthesized and also come into here, as well as being washed away in the plasma for the total body distribution. Well, we'd have to have a capillary picking it up, taking it to the venous system. But also diffuse in here and bind to a protein and also contribute to sperm development. So here we see those capillaries very nicely. So I always urge you to try to picture the molecule, where it goes. So the LH diffuses out, and that gets close to these Leydig cells and bumps into the receptor, promotes testosterone synthesis. As soon as it's synthesized, diffuses out. Here's that connective tissue capsule of the seminiferous tubule. So diffusion in all directions, washed away by the capillary, delivered to the body. There's also peptides that are secreted, and we picture them being washed away in the capillaries, returning to the pituitary ultimately to contribute to negative feedback. The testosterone targeting peripheral tissues and organs to have its effect. And then the female gonad, of course, will go into great detail of all the different cell types that are targeted by FSH and LH, and it is much more sophisticated than regulation in the male. So we gave the male gonad its time now. The female gonad will get much more detailed information emerging from it and time devoted to it when we discuss the female HBG axis. Well, we're getting very close to the 50-minute time point. What I will do is I'll record this lecture on the hypothalamus and post it as a separate lecture. It will be recorded just from a desktop. You won't see me in the little picture in the picture here. So I'll record that and post it for you to view on your own time. And then when I come to the classroom next, I'll pick up with lectures on Chapter 5. Appreciate your time and attention, and for those of you here today, hope you have a good weekend. Also, check the calendar for the posting of the first exam. The first exam covers the material that is within chapters 1 through 4, and I'll make sure that the exam is available long enough for you to have an opportunity to view the streaming video lecture on the hypothalamus, the slides remaining.